Hi, my name is Lauren Geek, and I am here to present the findings of our paper entitled Computer Simulation and Mathematical Modeling of the Interactions between Ecological and Sexual Selection to reveal the mechanism of sympatric speciation. The focus of our lab is to use these methods of computer simulation and mathematical modeling to unpack some of the mystery behind the evolutionary mechanism of sympatric speciation. Sympatric speciation is classified as speciation without geographical barriers. This is in direct contrast to allopatric speciation, defined as speciation with geographical barriers. To discuss the current understanding of sympatric speciation, let's use this example of a group of fish in a lake. All fish are feeding on this common food source pictured below. We can see the phenotype distribution of the fish is represented by this bell-shaped curve. On one end of the distribution, we have skinny fish, and on the other end, we have rounder fish. Now, in disruptive ecological selection, there are two different food sources, as pictured here. One is blue, and one is red. The skinny fish are adapted better to feed on the blue food source, while the rounder fish are adapted better to the red food source. In this instance, the phenotype distribution is less selective for the intermediate hybrids, as there is no food source best adapted for these hybrids. After disruptive ecological selection occurs, the next step in the speciation process is assortative mating. For example, all the skinny fish feeding on the blue food source in niche A have developed a blue color, while the round fish feeding on the red food source in niche B have developed a red color. Now, the distinct color can play a role in mating, as the fish in niche A only want to mate with other fish of its own color, and likewise with the fish in niche B. Thus, reproductive isolation has occurred between the two populations, and this has reduced the production of hybrids. This hybrid loss drives sympatric speciation. This is the most controversial part of sympatric speciation, as researchers try to understand how non-random assortment of a mating trait can occur when there is gene flow between the two niche groups. Here on the right, you can see the flow chart for the computer application that we have developed to investigate the mechanism of sympatric speciation. It can be broken down into two parts of the life cycle: the ecological selection and sexual selection. We first utilized individual-based computer simulation under the following parameters for genotype. We assumed each individual has 3 gene loci for ecological alleles and 1 gene locus for mating bias alleles. When 2 individuals mate, they produce offspring via random assortment. Looking at the ecological selection portion of the flowchart, we can imagine each gene locus has 60 alleles, which can be plotted on a 3-dimensional genotype-phenotype map, with each axis representing a gene locus, as shown here. The ecological genotype of each individual can then be visualized by each red dot shown on the 3D graph. We can then create niche resources in the genotype-phenotype map shown by the green spheres here. In this top map, we can see 1,000 randomly distributed individuals, and if the individual's genotype is adaptive to one of the 2 niche environments, that individual has access to food resources and can survive until mating and producing offspring. Next to the 3-dimensional graph, you can see the same data visualized in a 2-dimensional histogram graph. As you can see with the next generation, eventually the individuals will congregate into either niche A or niche B. Since the individuals are still randomly mating, you can still see the production of hybrids here. With 3 gene loci, there are 8 possible permutations of the loci. 6 out of 8 of these are non-viable hybrids, demonstrated by the 6 circles outside of the niche resource groups located on the graph. Next, we will look at the second phase of the flowchart, sexual selection. After the individual survives to maturation by falling in one of the two niche resource groups, they move to the sexual selection phase of reproduction. To conceptualize this, we can imagine a speed dating meeting. Each individual meet by random encounter, and the mating success of each pair depends on the sexual bias allele carried by each individual. If they carry the same mating bias allele, either XX or YY, they are 100% compatible. If they carry different mating bias alleles, their mating success is determined by this variable alpha, which ranges from 0 to 1. They can range from 0% compatible to 100% compatible based on the value of alpha. If the individuals match during the speed dating round, they move into the pool to become mating parents and reproduce offspring. Those individuals who do not match go into a new speed dating round with the other individuals who did not match, and they have another opportunity to find a mating partner. They can try this a total of n times, as n is the number of matching rounds. Now, if an individual is more discriminative, or we can say more picky about their mate, this would mean that alpha is a lower number. If this is the case, it is less likely for them to find a partner in n mating rounds, and they will go home without the opportunity to reproduce. Therefore, n is a measure of assortative mating cost, and both alpha and n are variables that determine the strength of sexual selection. This is the general layout of our individual-based computer simulation. Each generation of the population goes through the ecological selection, then the sexual selection of the life cycle before producing the next generation. We can then plot the change in population dynamics after a certain number of generations, referred to as variable G. Here we can see the result of our simulation. Initially, with random mating, the individuals in niche A and B continue to produce non-viable hybrids. Over time, you gradually see the reproductive isolation of the two niche populations and the reduction of hybrid populations between the groups. The next step was to develop a mathematical model based on the results from the individual-based computer simulation. Here in the model, you can see the 4 phenotype groups. In this group, you have individuals adapted to niche A with the X mating bias allele. This is the same for niche A with the Y mating bias allele, niche B with the X mating bias allele, and niche B with the Y mating bias allele. All parameters in the model are normalized to all groups, so NAX, NAY, NBX, and NBY add up to 1. Here is the matching probability matrix, which shows the probability that each genotype encounters each other, which is represented by a vector. The matching probability is displayed next to the vectors here. So the probability of encounter for AX and AX is AX squared. For AX and BX is AX BX. All of the cells in this matrix will add up to 1. Now there are 2 important variables here. The first is alpha, which we have already seen. Alpha is the mating bias value. The cell seen here with the alpha symbol displayed is a reminder that the different mating bias alleles are dependent on the alpha value, ranging from 0 to 1. The second variable is F, and this is called the offspring return ratio, which shows the ratio of the offspring with the same genotype as the parent generation. The values of F can range from 0 to 0.5. For example, if the ecological genotype in niche A is AAA, and in niche B is BBB, then the hybrid offspring produced could be ABA or ABB, etc. These are non-viable hybrids. Therefore, out of the 8 permutations, only 1/8 of the offspring are viable with the same genotype as the parents. The value of F would be 1/8. and 6/8 of the offspring are non-viable hybrids. Once we determine the number of viable matching parents based on mating bias value alpha and the number n of matching rounds that they are allowed to go through, then we can calculate the offspring ratios that each genotype parent can produce. For example, AX and AX will produce offspring the ratio of 100% AX. For AX and BX, only 1/8 will be AX, 1/8 will be BX, and 6/8 will be non-viable hybrids. The ratios in each cell add up to 1. Because AX, BX, AY, and BY all add to 1, the sum of all ratios in each of the matrix cells add to 1, and therefore all the parameters are normalized. The unit offspring matrix also assumes that each parent mating pair can only produce one offspring. After finding the unit offspring ratio, this ratio can be multiplied by a factor to get the number of offspring per generation if a mating pair has more than one offspring per generation. So now we can break down what happened in our mathematical model during each phase of the life cycle. These top two groups belong to resource niche B, and the bottom two groups belong to resource niche A. The interaction between these two groups is determined by ecological and sexual selection. We can remember the variable f here determines the offspring return ratios, or the ratio of the offspring with the same phenotype as the parents, making them viable offspring. So the strength of ecological selection here is determined by f. Now, in the sexual selection phase, we see that these two groups have the same mating bias allele X here, and these have the same mating bias allele Y. Having the same mating bias allele means they are 100% compatible. Therefore, the interaction between these two groups is determined by variable alpha, the mating bias value, and n, the number of matching rounds. Thus, the strength of sexual selection, or the matching success of the individuals, is determined by these two variables. Now we can take a look at the interaction of these two genotype groups. We notice that NAX is larger than NBX. This means the larger group will suffer less reduction in population ratio because it starts off larger than NBX. If we factor in the hybrid loss from each group, we can see here that if NAX has a population of 0.6, the reduction in the size of the phenotype group is 0.63. However, since NBX starts with a population size smaller of 0.4, the same hybrid loss of 0.375 will have a larger reduction in this phenotype group, which is 0.94. Therefore, the result of the hybrid loss within the interaction of these two groups will continue to make the larger group NAX larger and the smaller group NBX smaller. In order to have the maximum amount of pre-mating reproductive isolation, we need the NAX group and the NBY group to be large, and the NBX and the NAY groups to get smaller. In this way, the majority of individuals in resource niche A will have the X mating bias allele, and the individuals in niche B will have the Y mating bias allele, maximizing reproductive isolation between the individuals of the two resource niches. In summary, our mathematical model shows that each interaction represented by the arrows here have population loss. The effect of this loss is to make the larger groups larger and the smaller groups smaller. In this example, NAX is the largest group, and therefore the opposite group, NBY, would also be larger to maximize reproductive isolation. This would make NBX and NAY the smaller groups. This process is mostly governed by ecological selection, shown with the green arrows, and sexual selection, shown by the red arrows. Both the ecological and sexual selection forces are complementary. A small ecological selection can be compensated by a larger sexual selection, and vice versa. The interactions shown by the diagonal lines are more complex because the offspring will be distributed equally due to the differences in ecological and mating bias alleles, and will have less of an impact on population size. With this being said, the main forces driving reproductive isolation are ecological selection and sexual selection, shown primarily by these green and red arrows. In this model, we noticed a phenomenon which we have called incumbent selection. For example, after a certain number of generations, we can say NAX and NBY are the largest groups of the four, and NBX and NAY are virtually eliminated. Unless the remaining groups are exactly equal in size, we will see that the interaction between groups NAX and NBY will drive the larger group to be larger and the smaller group to be smaller, until there is only one group left in the entire system. We call this incumbent selection, meaning the larger population can use its numerical advantage to eliminate the smaller minority population, despite the fact that there are available niche resources for NBY, causing them to have increased reproductive fitness. They can still be driven to extinction via this incumbent selection phenomenon. To prevent this from occurring, we have to make certain assumptions within our model as follows. We can assume the niche carrying capacity of each niche group is limited. Therefore, the larger group cannot indefinitely increase in size. Additionally, we are to assume the population ecotypes in each niche are able to produce enough offspring to saturate the carrying capacity. Therefore, the carrying capacity of each niche is fixed, and each generation, the ratio between each niche population is also fixed. Here we can see the nonlinear difference equations we derived to describe the dynamics of the model. We next developed a computer application to solve this set of nonlinear difference equations. Here is an example of the computer model we developed to solve the set of nonlinear difference equations. You can see all of the relevant variables here. For example, the NA ratio, variable F, and variable alpha. All the parameters here again are normalized. Here's the phase portrait that plots AX versus BX. Since everything is normalized, we know the ratio of individuals in niche A with the Y allele because AY is just 1 minus AX, and the same goes for BX and BY. As we change the parameters, we can see how the phase portrait changes. The blue dots are the initial starting populations. So here, after 100 generations, you can see how the population ratios of AX and BX change. In this case, they both converge to a fixed point shown here by red dots. As we continue to change the parameters, we can see here just a sample of the different patterns we see in the phase portrait as we change the different variables. We observed the following few patterns in the computer application. First, we observed this pattern we have called convergent, meaning to converge to a fixed point. It is important to note that this is in contrast to a divergent pattern that does not converge to a fixed point. The value of the fixed points are displayed at the top. Despite increasing the generations, we see that the starting populations converge to a fixed point. We can see that this red dot represents a stable equilibrium point where the ratio does not change. In order to have maximum reproductive isolation, we want NAX and NBY to be larger. Therefore, we want the red dot representing the fixed point to be as close to the bottom right corner here, or as close to 1 as possible. This shows almost all of the individuals in niche A have the X mating bias allele, and almost all of the individuals in niche B have the Y mating bias allele, representing pre-mating reproductive isolation. A second pattern we observed is divergent, meaning there is no fixed point possible. The initial populations move to the diagonal line seen here in the middle. They are then driven by sexual selection to the origin or to the far end of the line. This represents there is only one type of mating bias allele left in both niche populations. The intermediate pattern between the convergent and divergent patterns is shown here as partially convergent. The solid lines here represent the initial population moving to a fixed point, and the faint lines show the population that will move towards the diagonal line with no fixed point. We call the portion of the initial population that moved towards a fixed point the percentage of convergence. Here is the dynamic animation of the two systems. On the left, we can see the system is convergent. The diagonal line is where AX is equal to BX, and it is an unstable ridge, meaning any deviation from this line and the point will be driven to a fixed point. If a population falls on the diagonal line, sexual selection will drive the population towards the origin or towards the opposite end, where there is only one mating bias allele remaining in the population. This is due to the fact that sexual selection prefers the predominant type of mating bias allele. Now, in the divergent system here on the right, the system is divergent where the initial populations move to the diagonal line and are driven by sexual selection to either the origin or the far end of the diagonal line. Next, we used numerical methods to compute the fixed point curve for the changes in any one variable. For example, if we change only the variable alpha and keep the remaining parameters constant, we can plot the fixed point trajectory as the alpha variable changes. For alpha equals 0, we will start at point AX equals 1 and BX equals 0. As we increase alpha, the fixed point will continue across the curved line until alpha reaches the value here, 0.59895. Until above this value, no fixed point is possible and the system becomes divergent. We can then repeat this process for each individual variable and plot the fixed point curves for each variable. The fixed point curve for alpha and F are very similar, starting at the point here in the lower right corner and continuing to fan out. For variable NA, which is the niche carrying capacity ratio, we see that the fixed points look like parabolas which intersect with the fixed point curves for alpha and F variables here. So as NA moves away from the value 0.5, either larger or smaller, it is less likely to have fixed point convergence. We can also plot the fixed point curve based on the changes in different variables as seen in the three-dimensional plot here. This shows the regions where a fixed point is possible with the different values of alpha, F, and NA, and a different number of matching rounds. The different colors on the plot represent the percentage of convergence. We discover in this model that there appears to be a line here beyond which no fixed point is possible. As we decrease the number of matching rounds, this three-dimensional prism gradually shrinks towards a point where NA is 0.5, alpha is 0, and F is 0. Based on these results, we have come to the following conclusions in regard to conditions favorable to system convergence or the emergence of a fixed point. First, strong ecological selection where variable F is small is necessary. Second, strong sexual selection where variable alpha is small is also necessary. Third, we need a low assortative mating cost or a large number of matching rounds, variable N. Fourth, we need roughly equal niche sizes between niche A and niche B in this example. These results and conclusions are under the assumption that there are no viable hybrids produced by each generation. If we now assume there are viable niche food resources available for these hybrids, then we can assume these hybrids can mature and produce offspring. We developed computer applications to investigate what will happen to our phase portraits under these new conditions with viable hybrids. With ecological hybrids, the net result is to increase the value of F as hybrids continue to mate with each other and produce more parental genotypes. By increasing the value of F, the strength of ecological selection decreases and inhibits the convergence of the system, therefore inhibiting pre-mating reproductive isolation. Now in the example on the right, we investigate what would happen with the mating bias allele hybrids. In this study, we have only one gene locus with two mating bias alleles X and Y. In this example, the combined effect of 3 different gene loci resulting in an abundance of hybrids with varying degrees of mating bias are analyzed. We find that the existence of mating bias hybrids decreases sexual selection by increasing the variable alpha and inhibiting system convergence and pre-mating reproductive isolation. Under these conditions, disruptive sexual selection must occur to eliminate the mating bias hybrids. Now, based on our findings, the system will still converge to a fixed point. However, as shown below, we will still get a distribution of hybrids closer to each extreme. Therefore, the fixed points will be weaker and produce weaker reproductive isolation between the two niche groups. Based on these observations, we can conclude that system convergence is more likely when there are more ecological gene loci because the hybrids are non-viable in our model. Therefore, the more gene loci present, the lower the number of variable F. In contrast, system convergence is more likely when there are few mating bias gene loci that are present due to the possibility of mating by all individuals within the same niche. The fewer mating bias gene loci results in fewer hybrids produced and stronger reproductive isolation. To minimize hybrid production, we can see disruptive sexual selection within each niche in order to eliminate the mating bias hybrid production. One way to have this is to have what is referred to as a magic trait, meaning an ecological trait that is also a trait necessary for mating. Let us imagine an example with a population of birds with varying beak sizes. In this population, birds with a larger beak and birds with a smaller beak are each adapted to different niches. Now, if beak size is also used by these birds as a mating signal, then the ecological and sexual traits are coupled together, as the disruptive sexual and ecological selections are linked. This is referred to as the magic trait scenario and can result in system convergence, where, for example, niche AX and niche BY continue to grow larger to a fixed point, making pre-mating reproductive isolation easier. If you have a system that is globally convergent, a small mutant allele, in this case X, can invade the system and reach a fixed point. For example, if you have a population with only the Y allele, and somehow there is disruptive ecological selection and the values of F and alpha are changed, you achieve this globally convergent phase portrait in the system, then a small mutant allele from a population variable X can invade and produce pre-mating reproductive isolation between the two population niche groups. Here, we have a generation plot of what happens when a small population of a mutant allele invades. For example, an initial population of AX, which is represented by the blue line here, has a starting value of 0.0001. This small population can invade and after 35 generations can achieve fixed point polymorphism between population AX and BX. BX is shown by the red line here. The green line represents the ratio of hybrid offspring, and as you can see, the two populations converge to fixed points. Thus, the hybrid ratio decreases. indicating there is pre-mating reproductive isolation between the two populations, AX and BX. Next, we investigate how initial pre-mating barriers can be invaded by alleles with a higher mating bias and further increase reproductive isolation. In this first study, we investigated what would happen with 3 alleles at the mating bias gene locus, shown as X, Y, and Z, instead of the previous 2 alleles X and Y. In this case, we discovered after the X and Y system had reached fixation, a very small population of the Z allele can be introduced here at generation 200 in this example. If the Z allele has a higher mating bias, it will invade and go to a fixed point. What we discovered is you cannot have 3 mating bias alleles existing in a fixed point polymorphism. For example, if these are the 2 fixed points here in the X and Y system, this is AX and BX and AY and BY, the emergence of a Z allele with a higher mating bias will invade and eliminate one of the original alleles, in this case, the X allele. The invasion of the Z allele will bring the fixed point for the Y allele closer to the right-hand corner, representing stronger pre-mating reproductive isolation. This is done by minimizing hybrid loss as the fixed points move closer to the further corners of the plot. The implication of this example is that once a 2 allele system has reached a fixed point, additional mutations with a higher mating bias can continue to invade, strengthening reproductive isolation. Similarly, we can have a mating bias allele that can modify the mating bias variable alpha. For example, if we have an allele referred to as a meta-allele that can change alpha from 0.3 to 0.1, this graph shows that the AX and BX populations have stabilized, and at generation 200, the small population of the meta-allele can invade and go to fixation, shown by the green line. It will increase the fixed point polymorphism of the populations and increase the overall reproductive isolation. This is an example of coupling, as the initial mating bias barrier system is coupled to the meta-allele system, increasing mating bias values and subsequently increasing reproductive isolation. The last step was to verify that our mathematical model corresponds to the results of our individual-based computer simulation. And here you can see that the results of the mathematical model are shown by the solid line, and the computer simulation results are shown by the dotted line. The results both correspond very closely. Based on the findings of our paper, we can propose the 5-stage process for sympatric speciation. In step 1, a single species subsists on a single habitat. Because individuals within a single species mate randomly, sexual selection will favor the predominant type of mating bias allele, which is the low mating bias alleles, and drive the high mating bias alleles to extinction because they are less likely to find a mate as they are more selective. Step 2, disruptive ecological selection occurs and selects against less fit hybrids with no food resources available. Step 3, selection against the high mating bias alleles is reversed. The high mating bias alleles is favored to invade the system and create pre-mating reproductive isolation. The invasion of the high mating bias allele can minimize the intra-niche hybrid loss. In step 4, after the high mating bias alleles invade and establish fixed point polymorphism, this can then recruit other types of barrier mechanisms to come in and further increase the reproductive isolation. For example, the Bateson-Dobzhansky-Moeller mechanism of divergence tends to reduce gene flow only after there is a pre-established reduced gene flow between the two populations. In late-stage speciation, you have many types of barrier mechanisms that couple together to complete the speciation process, making it irreversible. Finally, in step 5, almost no gene flow between the two niche groups is present. Intraspecific sexual selection again favors low mating bias alleles and selects against high mating bias alleles, and the process returns to step 1. In summary, we have developed a simple mathematical model to investigate the mechanism of sympatric speciation. We have done this by analyzing sympatric speciation as nonlinear dynamic systems. Second, under certain favorable conditions, fixed points can appear in our system's phase portraits to create non-random assortment of mating bias alleles and pre-mating reproductive isolation. Third, all the parameters in our model are normalized, so they can be applicable to a wide range of situations. Therefore, we're able to produce conditions required for a mutant mating bias allele to invade and establish pre-mating reproductive isolation. Fourth, the next step for our group is to study how this initial pre-mating reproductive isolation can be coupled to additional pre-mating and post-mating barrier mechanisms to further strengthen and complete the speciation process. See our next video to see the results from our subsequent studies. Lastly, I want to thank everyone in our lab group and all of our collaborators.